welcome everyone to the inaugural Talk2D seminar presented by the ARC Research Hub for Advanced Manufacturing with 2D Materials, or AM2D. My name is Dr. Philip Sheath, and I'm the manager of the hub, and I'll be introducing the speakers today. Before we begin, AM2D and Monash University wish to acknowledge the people of the Kulin Nations on whose land we are gathered on today. We pay our respects to elders past and present. And I ask those of you online to pay respects to the traditional owners on the lands which you are situated. Today we have three diverse speakers who wish to tell you about their fascinating work. First up, we have Dr. Cécilia Ménard-Moyon. Cécilia is a research director at the French National Centre for Scientific Research, or CNRS, having been in that role since 2021. Cecilia's research interests include functionalization of carbon-based nanomaterials for biomedical applications, the self-assembly of amino acid derivatives and peptides, and the formation of hydrogels for on-demand drug delivery. It's my pleasure to hand over to Cécilia, whose talk is titled How to Control Chemical Derivatization of Graphene Oxide.
Thank you very much, Phil, for the kind introduction. Okay. Very good. So good morning, everyone. Thank you very much for being here in the room, and also thank you very much for those participating online. So that's great pleasure for me to talk to you today about the chemical derivatization of graphene oxide, and I will also show you some of our results on the biomedical applications of graphene oxide. So I will start with an introduction on the chemistry of graphene oxide, what are the main issues and challenges. Then I will present you some strategies for the monofunctionalization of graphene oxide and the double functionalization of graphene oxide. And then I will show you two short examples on the biomedical applications of graphene oxide. So just a few words about my lab. So my lab belongs to CNRS. So CNRS stands for National Center for Scientific Research. It is located on the campus of the University of Strasbourg. So this is France. Well, Paris, sorry, I don't have laser pointer yet, but well, Paris is in the north part of France, and Strasbourg is in the red circle, so it's on the east-north part of France, very close to Germany. So you can see Strasbourg is a beautiful city with a strong German style. The University of Strasbourg is very attractive. We have many foreign students. We have not 4, but 5 now Nobel Prizes still in activity. So this is my institute, the Institute of Molecular and Cellular Biology, in which there are 3 different labs, in particular my lab, immunology, immunopathology, and therapeutic chemistry. So my research interests are mainly focused on the development of multifunctional nanoparticles for applications in nanomedicine. I'm interested by inorganic nanomaterials and organic nanoparticles. So among inorganic nanomaterials, I'm mainly focused on different types of carbon-based nanomaterials like carbon nanotubes, small size carbon nanoparticles like carbon dots and graphene quantum dots, graphene, also other 2D materials beyond graphene like molybdenum disulfide, and also more recently magnetic and plasmonic nanoparticles. I'm also interested in the self-assembly of amino acid derivatives, for instance, to form spherical nanoparticles, also peptides to control the growth of nanotubes, and also hydrogels made of the self-assembly of amino acid derivatives. Thank you very much for the laser pointer. And my main objective is to control the synthesis and the chemistry that we do on these different types of nanoparticles for various biomedical applications like therapy, imaging, diagnosis. And also in parallel, we are evaluating their potential toxicity, their pharmacology, and their biodegradation. So Well, you probably know that graphene oxide has many interesting properties that can be exploited in a variety of fields. For instance, in lithium batteries, as supercapacitors, solar cells, membranes for water filtration, sensors, catalysis, and also in the biomedical field. For instance, as a drug delivery system for bioimaging or as a biosensor. So in the group, we are exploiting graphene oxide as a platform for drug delivery. Why? Because graphene oxide first has a very large surface area. It has also a high water dispersibility because it is composed of a variety of oxygen-containing groups. And also these groups give the possibility to, for derivatization, to control the conjugation of bioactive molecules on graphene oxide. But when we started to work on the functionalization of graphene oxide, actually we faced many issues. And this is related to the fact that graphene oxide has different oxygenated groups on the surface having a rather high chemical reactivity. So there is a risk of unwanted multiple simultaneous reactions. In addition, some reactions might require heating, but graphene oxide is thermally unstable. So the material can be reduced to different extents depending on the temperature. So in this context, our objectives are to control the covalent functionalization of graphene oxide using reproducible reactions that can be performed in mild conditions, allowing to preserve the structure and the chemical composition of the material. And also one of our objectives is to characterize the functionalized graphene oxide unambiguously, because you will see that this is very challenging. So the structure of graphene oxide, in fact, is not fully elucidated. It is still under strong debate. In the literature, there are different types, different structures of graphene oxide that have been reported. So here I've selected two of them. So the first one is the Lev-Klinovsky model reported in 1998, in which the basal plane of graphene oxide is mainly composed of epoxides and hydroxyls, while on the edges there is a low amount of carboxylic acids and also other types of oxygenated groups. The second model I selected is the Dekani model reported in 2006. So also in this case, the basal plane is mainly composed of epoxides and hydroxyls, but in this case, the edges do not possess carboxylic acids. So you see that there is an ambiguity regarding the presence or the absence of carboxylic acid groups. And this is something that we have tried to elucidate. So now I would like to present you different strategies for the monofunctionalization of graphene oxide by exploiting either the epoxides or the hydroxyls, because they are present in a very large amount on the surface of the material. And first, let's start with the reactivity of graphene oxide towards amines that are highly nucleophilic compounds. So in the literature, the main reaction which is used to functionalize graphene oxide is the amidation reaction. So this reaction takes place between the carboxylic acids on graphene oxide and an amine derivative in the presence of coupling agents. So coupling agents are necessary to activate the carboxylic acids, so giving the formation of this amide bond. But in fact, in the presence of an amine, the main reaction occurring is the epoxide ring opening. As I mentioned before, epoxides are present in a large amount, and they are highly reactive towards amines. So in fact, in these conditions, there are two reactions occurring, so the epoxide ring opening and the amidation reaction. So the epoxide ring opening is a reaction which is very fast, actually. It can occur at room temperature, in water, so in very mild conditions, and there is no need of catalyst. But the main issue is that in the literature, there are many articles reporting the amidation of graphene oxide without taking into account the possibility of this concomitant epoxide ring opening. So this leads to false and incomplete structures of functionalized graphene oxide. So we investigated more in detail this reaction. So we performed first the amidation reaction, and also in the concomitant epoxide ring opening. So using this diamine derivative with one of the amine protected with the Bock group here, we used EDC and HOBT as coupling agents. The second step was the cleavage of the Bock group to have ammonium functions that we exploited then to assess the level of functionalization using a colorimetric test. In parallel, we also performed the same reaction, but without any coupling agents. So in this case, only the epoxide opening occurs. We assessed the level of functionalization using the colorimetric cassette test, so allowing to determine the amine loading, which was in a range of 240, 275 macromole of amines per gram of graphene oxide in both cases. So you see that there was not much difference in terms of amine loadings between the two different conjugates. So meaning that if ever there are carboxylic acid groups on the edges of the material, then they have a very low reactivity or no reactivity, or simply maybe that there are no carboxylic acids. So remember that when we mix graphene oxide with an amine, the main reaction occurring is the epoxide ring opening, with or without coupling agents. We characterize the graphene oxide and the functionalized graphene oxide by solid state NMR, in particular using the magic angle spinning NMR. So in black, you can see the carbon 13 NMR spectrum of the starting graphene oxide. So there are three main peaks. This one at around 60 ppm assigned to the epoxides. This one at around 70 ppm for the hydroxyls, and the carbon-carbon double bonds here at around 130 ppm. You can see here that in this area, there are no peaks corresponding to the carboxylic acids. After the reaction, so we can see first peaks here appearing corresponding to the CH2 of the amino linker. And most importantly here, a decrease of the peak corresponding to the epoxides. So confirming the epoxide ring opening. We then characterized the graphene oxide by solid state NMR and using quantitative carbon 13 NMR. So we could determine that graphene oxide was composed of around 1/3 of epoxides, 1/3 of hydroxyls, and 1/3 of carbon-carbon double bonds. And we could see that there were 3.5% of carboxylic acid, so very low amount. We then extended the characterization to other sources of graphene oxide. So this was graphene oxide from the NanoInnova Spanish company. So we also characterized another commercially available graphene oxide from Antolin Spanish company. And we found that there were 2.5% of carboxylic acids. And a graphene oxide prepared by the group of Costarelos in Manchester, having 2.9% of carboxylic acid. So you see that among these different sources of graphene oxide, the percentage of carboxylic acids is in a range of 2.5, 3.5%, which is very low, as expected. So to functionalize graphene oxide, while it is much better to exploit the epoxides that are present in a larger amount on the surface of the material. So now that we have investigated the reactivity of the epoxides on graphene oxide, we studied different reactions to derivatize the hydroxyls on the material. And the first reaction is the Williamson reaction, which is an esterification reaction, so allowing to form an ester bond here. So this reaction takes place between the hydroxyls on graphene oxide and this iodinated compound in the presence of a mild base, so potassium carbonate. Then the last step was the cleavage of this Bock protecting group, allowing to have ammonium groups, helping to assess the level of functionalization using the cassette test. And this is what we did here. And we optimized the reaction conditions to reach higher loadings. And in particular, we increased the temperature from room temperature to 80 degrees. In fact, we could see that the best conditions allowing to have the highest amine loading were those performed at room temperature. So we characterized by XPS the different functionalized graphene oxide. And in particular, we looked at the peak assigned to the CO bond and the CC bond. And we could observe that when we increased the temperature of the reaction, there was a significant decrease of the CO bond. And this is, this indicates that there was a partial reduction of graphene oxide upon heating. And this was confirmed by a thermogravimetric analysis that we performed under nitrogen atmosphere. In black, you can see the thermal profile of the starting graphene oxide. When we increased the temperature of the reaction, there was a lower weight loss, so confirming the partial reduction of graphene oxide upon heating. So you can see here that it was not necessary to do the reaction under heating because there was a partial reduction of the material, and anyway, we could not reach high loading. So it is much better, again, to perform reactions at room temperature. The second reaction that we explored is the esterification reaction, so taking place between the hydroxyls on graphene oxide and this carboxylic acid derivative linked to terminal amine protected with the Bock group. So allowing to form this ester bond. So this reaction was performed in the presence of coupling agents to activate the carboxylic acid here. And the last step was the cleavage of the Bock group to have the ammonium moieties, allowing to assess the level of functionalization. So to confirm the occurrence of the reaction, we used solid state NMR. So in red, you can see the spectrum of the graphene oxide, the starting graphene oxide. After the reaction, so we were delighted to see peaks here corresponding to the CH2 of the linker, and most importantly, to see this peak here, so close to 190 ppm, confirming the presence of the ester bond. And also, what we can notice is that the peaks of the epoxides and hydroxyls were unchanged after the reaction, so showing that here the conditions were mild and did not affect the chemical composition of graphene oxide. So now that we have investigated the reactivity of the epoxides and the hydroxyls on graphene oxide, we then combined two different reactions to selectively functionalize graphene oxide and design strategies of double functionalization. And first, we combined the epoxide opening with this diamine derivative, as I mentioned before, with the esterification of the hydroxyls using this carboxylic acid derivative linked to this amine protected, in this case, with photo-cleavable protecting group, which is orthogonal to the Bock group in a sense that we can cleave one without affecting the other. And this protecting group, this photolabile protecting group, is interesting because it has a nitro moiety that actually facilitated the characterization of the material and confirmed the occurrence of the reaction, in particular by XPS, because when this is the XPS survey of the functionalized, the double functionalized graphene oxide. And looking at the N1S peak here, there were two main peaks. So this one at around 400 electron volts, assigned to the amines and carbamates. And most importantly, this one assigned to the nitro moiety, so confirming the presence of this nitro group on the surface of graphene oxide, so confirming the outcome of the reaction. The second strategy for the double functionalization of graphene oxide is this one, so combining again the epoxide opening with the hydroxyl derivatization. And for the epoxide ring opening, we used not an amine in this case, but a thiol containing molecule, because thiols are also highly reactive towards the epoxide. They are highly nucleophilic. So this first reaction led to this conjugate. And then the hydroxyls were derivatized with benzoquinone. So benzoquinone has two double bonds that are highly reactive towards nucleophiles. So we could derivatize the hydroxyls in very mild conditions at room temperature. And then this double bond was highly reactive towards anamine, and we used this phenylalanine derivative because, in fact, it has SF5 moiety that could facilitate the characterization by FTIR and by XPS, as I will show in the next slide. And also, it has a carboxylic acid here that we can further exploit for derivatization with molecules of interest. So, regarding XPS, so this is the survey of the starting graphene oxide. And when we look at the S2P area, there is one peak assigned to the organosulfate groups, because there are organosulfates on the surface of graphene oxide present in a low amount. After the first reaction, so after the epoxide ring opening, we could see the appearance of nitrogen. And looking at the S2P area, there was a second peak appearing assigned to the S carbon bond, so confirming the occurrence of the reaction. And most importantly, so looking at the XPS of the final conjugate, so there was the appearance of the F1S peak thanks to the SF5 moiety here. And in the S2P area, a third peak appeared assigned to the SF bond. So again, confirming the presence of this functional group on the surface of graphene oxide. So you see that XPS allowed to confirm the occurrence of the two successive reactions. And also FTR was very useful because the SF switching peak is very specific and is in this area, as you can see for the FTR spectrum of the molecule alone, the phenylalanine derivative. And we could find this peak only in the FTR spectrum of the final conjugate. And we also exploited the fact that benzoquinone is present on the surface of graphene oxide because it has the redox activity. So we used cyclic voltammetry. So this is the profile of graphene oxide with no specific redox activity. This is the profile of benzoquinone with these two characteristic peaks that we also found in this intermediate conjugate and also in the final conjugate, so confirming the presence of benzoquinone on the surface of graphene oxide. So you see that by exploiting different characterization techniques, we were able to confirm the outcome of the two reactions on graphene oxide. So to conclude on this part, well, I have shown you that in the conditions of the amidation reaction, there is the concomitant epoxide ring opening, which is in fact the main reaction taking place. We have shown by solid state NMR that the carboxylic acid groups are present in a very low amount. We have also designed different strategies for the double functionalization of graphene oxide by combining epoxide ring opening and the derivatization of the hydroxyls. There is also the possibility to further derivatize the graphene oxide by exploiting the amino groups and carboxylic acid groups that we grafted on the surface of graphene oxide. And it is something that we have explored for the conjugation of bioactive molecules. And well, I want to stress also that well, you've seen that the characterization of functionalized graphene oxide is challenging. So we have to use combination of different techniques like NMR, thermogrammetric analysis, colorimetric test, XPS, FTR, cyclic voltammetry. And what we did is that we used well, tagging molecules that can facilitate the characterization by one or few of these techniques. So now I would like to switch to the second part of my talk, which is more related to the biomedical applications of graphene oxide. And first, I would like to show you some of our results on the enzymatic degradation of graphene oxide. Because if we want to exploit graphene oxide for biomedical applications, in particular for in vivo applications, we need to know what is the fate of the material, or how the material can be degraded in vivo. In fact, we have shown that peroxidases are enzymes that can degrade graphene oxide. So they are human enzymes like myeloperoxidases, peroxidase. And also in this study, we used a plant peroxidase as a model enzyme, so the horseradish peroxidase. Here, our objective was to see if we could accelerate the degradation of graphene oxide by conjugating substrates of the enzymes on graphene oxide. So, for instance, coumarin here and catechol. So, both are very good reducing substrates of peroxidases. So, the objective is to attract somehow the enzyme in close proximity to the material and thus facilitate, accelerate the degradation of graphene oxide. In addition, catechol is known to be a good redox mediator in a sense that it can favor electron transfer and prevent enzyme inactivation. So we prepared these two conjugates that we incubated with the enzyme in the presence of hydrogen peroxide to activate the enzyme for a period of 20 to 30 days. We also used this control, an amino graphene oxide with no specific reactivity. So we monitored the degradation by transmission electron microscopy, also by Raman spectroscopy, but today I will show you only the TEM results. So for graphene oxide, so this is the graphene oxide before the treatment. And at the end of the treatment, after 30 days, we could see sheets that were partially degraded. So you see some holes in the sheets. While already after 20 days for the sample of graphene oxide functionalized with coumarin or with catechol, we could not see any sheets on the TM grid. We could see only debris. So showing that the degradation was conclusive and was enhanced. While for the amino graphene oxide used as control, the degradation was not uniform in the sense that there were sheets that were degraded or partially degraded and some that were not degraded. So you see here that by proper derivatization, we could enhance the degradation because we could somehow favor the interactions between the graphene oxide and the enzyme. So now to finish, I would like to show you how we exploited double functionalization of graphene oxide for cancer therapy. So the idea here is to exploit graphene oxide as a platform, so to graft targeting ligand of cancer cells, so to make the treatment specific, and also to graft a photosensitizer. So photosensitizer is a molecule that is able to form reactive oxygen species like singlet oxygen or hydroxyl radicals under light irradiation. And these reactive oxygen species are highly toxic for the cells. The idea also was to exploit the photothermal capacity of graphene oxide because graphene oxide under irradiation with near-infrared light can absorb the light energy and convert it into heat. And this can be used to treat tumor. So we wanted to combine both therapies, so photothermal therapy and photodynamic therapy, to treat cancer cells. So we prepared this double functionalized graphene oxide. So in this case, actually, we used the epoxide ring opening and we mixed the graphene oxide with two different amines. So we could conjugate folic acid, which is a targeting ligand of cancer cells because it can bind to the folate receptor, which is overexpressed on the surface of cancer cells. And we also conjugated chlorine E6, which is a photosensitizer, so allowing to generate reactive oxygen species under light irradiation. So this is what we did. We incubated the double functionalized graphene oxide with cancer cells for two different incubation time. So in green, you can see viable cells, so that was stained with a specific dye, and in red, the dead cells. So these are the cells incubated with the double functionalized graphene oxide without any light irradiation. So we see many viable cells. When we performed the single treatment, so photothermal therapy by irradiating the cells with a laser at 808 nanometers, you can see much less cells and also some dead cells. This is also something that we could see, but to a much lower extent when we performed the single photodynamic therapy by irradiating the cells with a laser at 60 nanometers, so allowing to excite the photosynthesizer and generate the reactive oxygen spaces. When we combine the two treatments, then you can see that here there were much less cells. And we quantified the number of cells using a specific assay to determine the cell viability. So to make it short, let's look at the 8-hour incubation time. When we performed the single photodynamic therapy, you can see that the number of viable cells decreased. It decreased even more when we performed the single photothermal therapy. And when we combined the two, you can see that the cell viability was even much lower. So indicating that by combining the two laser radiation to perform the double therapy, we could induce a much higher cell mortality. So now the next step is to perform in vivo experiments to treat tumors. So this is the end of my talk. So I would like to acknowledge the team of Alberto Bianco here, in particular those people involved in this work. I would like to thank some of my collaborators, so Jesus Raya for NMR experiments in Strasbourg, Costarelos for providing graphene oxide, Yuta Nishina for also providing graphene oxide and for cyclic voltammetry experiments. I would like to thank as financial support CNRS, so in particular because I'm here in Australia thanks to a specific CNRS grant with a group of Kurosh Kalantarzadeh. I'd like to thank the European Commission, in particular the Graphene Flagship, regional funding, and also thank you very much you for your attention in the room and online, and I'll be happy to answer your questions.
So those of you online, feel free to unmute yourselves and ask a question if you have any questions.
Mm-hmm. Thank you.
Yeah, they won't hear. Thank you so much for the great talk. My question was regarding the degradation studies of graphene oxide that you did. I think that also kind of correlates with the safety of graphene oxide when it enters human's body. And I had a couple of questions about that. First is there are also some debates about safety of graphene oxide based on its size. So very small particles of graphene oxide compared to big sheets, they behave differently. And also the oxygen level, like if you have a reduced graphene oxide or partial reduced graphene oxide or graphene oxide, how different they behave in terms of degradation.
So to answer the first question on the safety, more or less, of graphene oxide and the impact of the dimension of the material. Well, so we have shown that, so in collaboration with a group of Costarelos, that while graphene oxide with a rather small size, for instance, let's say below 500 nanometers in terms of lateral size and even much shorter, so can be, are biocompatible, no specific toxicity. So there were various studies that were performed in this group. In particular, that the material also can be eliminated from the body and in particular in the urines. We have also shown in collaboration with a group of Costarelos that the thickness of the material is also critical in the sense that, well, monolayer, so per definition, graphene oxide is monolayer, but some samples and commercially available samples may be a few layer graphene oxide. So monolayer graphene oxide can be also easily eliminated from the body, while thicker graphene oxide can accumulate in some organs, and this may be critical. So yes, the size, the thickness is really are crucial parameters. I would say the level of oxygen also is very important. For instance, related to the degradation of the material, we have shown that. So we have actually performed a comparative study with 3 different sources of graphene oxide with different dispersibility rate, I would say. So with samples having a very high water dispersibility and others that tend to aggregate and have a much lower water dispersibility. And we have seen that the graphene oxide with the highest water dispersibility was the one that was the most efficiently degraded by amylo peroxidase. So it really has an impact on the biodegradation and also on the biocompatibility, because graphene oxide with a much lower content of oxygens, for instance, reduced graphene oxide, is less dispersible in water, so it will tend to aggregate, and then the interactions with cells are different, and the fate of the material might be different also in terms of degradation and elimination from the body. It's very difficult to generalize, but this is somehow a tendency.
Really great talk. I mean, say some of the students will benefit greatly from learning about the chemistry you're doing. So thanks for that. I have a question about the cancer therapy. Now, many years ago, when people were working with nanotubes, they started to think about it using nanotubes as a cancer, therapy, because you could use the infrared like what you're doing. Now, how different would a graphene oxide based your therapy be different from the nanotube based work? I'd like to hear your thoughts. And also, I guess, how does it compete with existing technologies? I mean, there are a lot of drug carriers, that kind of materials. So what is the point of difference?
Thank you very much for your question. So it's very broad. So, I mean, we could talk for hours for that, but I will try to make it short. So I would say first that there is no universal drug delivery system, for instance. I will not say that graphene oxide is better than carbon nanotubes or better than gold nanoparticles, for instance, for photothermotherapy. Well, I would say that in terms of, for instance, photothermotherapy, graphene oxide, carbon nanotubes, so carbon-based nanomaterials, are very competitive compared to gold nanoparticles. Then, if we compare carbon nanotubes and graphene oxide, well, I would say that, of course, they have similarities because they are carbon-based materials. Okay. Then they are also distinct, in particular in terms of morphology. So, for instance, with carbon nanotubes, there is the possibility to exploit the internal cavity for encapsulating drugs, things that we cannot do with the graphene oxide. But with graphene oxide, the surface area is higher, so it's possible to reach higher loadings of drugs. Then they interact differently with cells. I would say carbon nanotubes really they thanks to their tubular morphology, they cross biological barriers in a very efficient way. But somehow the main limitation also is that we have faced some issues in some projects with the targeting capacity of the nanotubes because they penetrate very efficiently in cells. So sometimes it's very difficult actually to be specific for this part. While, for instance, with graphene oxide, we have shown that, it was shown in a group that they can interact specifically with some cells, for instance, with monocytes. So this can give some opportunities for the treatment of some diseases, for instance, leukemia. So, it's difficult, as I mentioned to you, to say, well, this one is better than the others. It's just that we try to exploit their intrinsic properties because they have also intrinsic properties for imaging. Try to exploit their specific morphology, and then also it depends on the targeted biological applications. I mean, the objective being to somehow be able to treat very hard to treat diseases for which there is no specific treatment, because there is always a balance between benefit and risk, of course. Okay.
Thank you very much, Cécilia, and please give Cécilia another thank you.